When a microscopic hazard successfully breaches the human airway, the initial localized damage sets off a biological chain reaction that rapidly threatens the entire host. By tracking the cellular response to these threats, we map a predictable 10-step pathophysiological cascade, converting a localized pulmonary insult into complete systemic collapse. First, alveolar epithelial and endothelial cells govern gas exchange, produce surfactant, and maintain barrier integrity. Second, the mechanical diaphragm drives ventilation. Step 1 reveals diverse insults attacking these systems, including pneumonia, sepsis, aspiration, and toxic inhalation. These hazards can originate locally, like inhaled toxins, or systemically in the blood, like pulmonary ischemia. Whether the hazard breaches from the outside or from within, the respiratory system processes every one of these diverse threats through an identical structural vulnerability. Step 2 begins with a sudden plunge in the production of cellular ATP. ATP is the primary energy-carrying molecule that powers biochemical processes in the body. Inside the alveolar epithelial cells, this energy shortage immediately cripples normal oxygen handling and disables essential membrane transport mechanisms. Simultaneously, a parallel crisis strikes the diaphragm. The sudden ATP deficit causes immediate critical fatigue in the respiratory muscle fibers. This severe drop in cellular energy causes step 3, pump failure. Healthy alveolar epithelial cells continuously operate microscopic pumps to actively transport sodium out of the air spaces, which pulls water with it and keeps the alveoli completely dry. Without ATP to power them, sodium transport halts. The tissue loses its ability to clear fluid, and protein-rich edema begins leaking from the capillaries, rapidly flooding the alveolar space. This barrier leak physically blocks oxygen from reaching the bloodstream. The primary cause of hypoxia here is losing the dry environment required for gas diffusion. As cellular control degrades, step 4 begins, calcium dysregulation. Without energy to maintain gradients, intracellular calcium levels spike dangerously high. This calcium overload heavily degrades epithelial and endothelial barrier function, activates destructive cellular proteases, and triggers severe bronchospasm in the surrounding smooth muscle. Step 5 accelerates the damage through the mass production of ROS, or reactive oxygen species. These are highly unstable oxygen molecules that strip electrons from cellular structures, destroying them. These reactive molecules flood the tissue, generated by infiltrating neutrophils, by failing mitochondria, and sometimes high-concentration clinical oxygen. The downstream damage is severe: structural injury to the epithelium and endothelium, coupled with severe dysfunction of the surfactant necessary to keep alveoli open. The biological environment becomes highly acidic in step 6. Widespread systemic hypoxia forces the oxygen-starved body to switch to anaerobic metabolism, which produces heavy amounts of lactate. At the same time, the diaphragm fatigue established in step 2 causes ventilation to fail. The body cannot exhale efficiently, leading to dangerous carbon dioxide retention and severe respiratory acidosis. Calcium overload, ROS, and combined acidosis lock pulmonary cells into a self-reinforcing cycle of destruction that escalates long after the initial insult passes. In step 7, the immune system detects this widespread cell damage and triggers massive inflammation by releasing a storm of cytokines. Cytokines are signaling proteins that coordinate the body's aggressive immune response. One specific cytokine, IL-8, plays a critical role here. It aggressively recruits massive swarms of neutrophils out of the bloodstream and directly into the lung tissue. This intense inflammatory response completely breaks down the remaining cellular barriers, resulting in profound capillary leak that drowns the tissue in protein-rich fluid. The body mounts a desperate, macroscopic physiological defense in step 8, compensation. Systemically, the nervous system activates. The patient develops tachypnea, a rapid breathing rate, and begins recruiting accessory neck and chest muscles to force air into the lungs. Inside the chest, the lungs initiate hypoxic pulmonary vasoconstriction. This is a physiological reflex where pulmonary arteries clamp down, attempting to redirect blood flow away from flooded tissue and toward regions that can still exchange gas. The compensation strategy relies on forcing a deeply fatigued diaphragm to work exponentially harder. Eventually, the muscle fails completely. The microscopic cascade finalizes in step 9, organ failure pattern. Clinically, this manifests as acute respiratory distress syndrome, or ARDS. ARDS causes diffuse alveolar damage, transforming soft, compliant lungs into severely stiff, fluid-filled structures. The patient experiences refractory hypoxemia. This is a state of dangerously low blood oxygen that continues dropping, resisting even high-concentration oxygen therapies. The diaphragm goes completely flaccid, and systemic CO2 spikes to lethal levels. In step 10, surviving cells face necrosis or fibroproliferative remodeling, becoming permanent scar tissue. This cascade highlights the respiratory system's defining vulnerability. In organs like the liver or kidney, local tissue death typically remains contained to that structure. But in the lung, cellular damage means a total halt to gas exchange. The inability to oxygenate the blood starves the entire body. A localized chest injury inevitably forces every other organ into complete systemic failure.